It has been a long way to this final week. Hi, I'm Han Jung-suk. In this week, we learn about the practical aspects of a color. As a start, we review some color order systems in order to communicate the color in a proper way. Today, we begin with the quantitative color systems. The quantitative color systems were built up based on the color measurements. However, when you look into the quantitative measurement of a color, the psychophysical aspect, such as how the human perceives a color, was very critical to quantify the color. Among the various types of quantitative color systems, we learn about only two. One is XYY or YXY, and the other is CED. The XYY includes the color of direct light as well as the color of reflected light. The reflected light is, in another word, object color. But when you need to describe the object color, you better adopt the CED system. Okay, let's begin with the XYY system. The large Y here indicates the luminance. So the large Y refers how bright a color is. For example, how bright a bulb is or how bright a piece of paper is. Then the small x and small y. These should be written in lower cases. They indicate an XY coordination of a color in a two-dimensional space. The coordinates contain the chromaticity information. So the XY space should contain various colors. As displayed in this chart, indeed, you can find any color you have ever seen in your life. All of them are within this horseshoe-shaped diagram. This is called as a chromaticity diagram, or more formally, the CIE 1931 chromaticity diagram, because the diagram was proposed by the CIE in 1931. Along the border of the diagram, you can find the wavelengths of visible light. And the purple line is created here, which tells that the magenta is not the color directly refracted from white light. There is no indication of a corresponding wavelength here. So the colors on the purple line are resulted through the additive mixture of more than two colors. Then where is the large Y? The Y exists in the direction of Z-axis. In the chromaticity diagram, the color properties are basically defined in terms of X and Y coordinates. The second way is referring the dominant wavelength and the purity. For example, the color sample 1 has the dominant wavelength of 550 nanometers. You can find the dominant wavelength by extending the connection line between the white point and the sample position toward the border of a chromaticity diagram. Secondly, the purity is approximately 50% because the distance from the white point is almost the halfway to the border. Therefore, the purity of sample 1 reaches approximately 50%, and a purity of a color can reach up to 100% when the colors are positioned along or anywhere on the borderline. In case of sample 2, here, it's not possible to define the dominant wavelength because the extension from the white point reaches to the purple line. In this case, you can mention the complementary dominant wavelength when the extension is extended to the other direction. Another utility of the chromaticity diagram is about color mixture. when we add sample 1 and sample 2, the mixed light will be on the line that connects sample 1 and sample 2. Depending on the intensity of each, the exact place will be determined. Therefore, we can easily identify an area of color reproduction of a display that generates colors based on RGB primaries. When we just need to know where the R, G, and B primaries are located, then the mixed results of the RGB are within the triangle. That is called color gamut. Different displays have different sizes and shapes of color gamuts. Do you remember this slide from last week? Yes, the temperature of a black body, as it is heated through its range of color, can be graphed as a line running through the chromaticity diagram. The Planckian locus is presented typically on this chromaticity diagram. Besides, a number of illumination standards are regulated and documented based on the chromaticity diagram. Then where is this XYY from? I tried to summarize the procedure as compact as possible. First, the large Y is one of the large X, Y, and Z. Each of X, Y, and Z is calculated by multiplying the power of spectral properties of sensed light along the visible light with X, Y, or Z three stimulus functions here. Each of the X, Y, and Z function is derived through the computation of R, G, B three stimulus functions. The initial R, G, B three stimulus functions were formulated based on the color matching experiments using three primaries. Going back to the XYY, the small x is actually the ratio of large X, and the small y is the ratio of large Y, respectively. The entire procedure looks quite complex, but in fact, pretty much systematic. For those who wish to know more about this, I recommend the book Principles of Color Technology by Dr. Behrens. One disadvantage of the XYY is that the steps and spacing within the diagram are not visually uniform. In 1976, the CIE, the International Commission on Illumination, proposed the CIE 1976 LAB system. The CLAB system attempted to maintain the perceptual uniformity within the space. In the CLAB system, the L indicates the lightness, and this varies between 0 and 100. Then, the A and B axis define the hue characteristics of a color. Axis A defines how reddish or greenish a color is, and axis B defines how yellowish or bluish a color is. This is a messy desk where I used to work. Let me measure the L values of various whites here. The L value of white displayed on monitor as 100 as the mixture of maximum values of RGB. And this white is direct light. However, the white paper, white telephone, and white wall are reflected lights. As we learned, the color of a reflected light is dependent on the light source. Therefore, I need a spectrophotometer. My spectrophotometer emits the standard illuminant, D65, over the paper. Then it receives the reflected light from the surface of the paper and then converts it into the LAB values. In terms of L values, the 4 white should have almost the same lightness. However, if I measure the 4 whites using a luminance meter, it is a different story. A luminance meter measures the luminance. The unit is candela over a square meter. The candela tells us the power emitted by the light source. In the previous slide, the L values of 4 white colors were almost equal to 100. However, in terms of luminous intensity, the monitor emits at least 5 times more powerful luminous intensity than the paper reflects. Therefore, the lightness of L of a CED system is the relative value. Regarding the uniformity of the perceptual distance of a color in the CED, actually it is not yet perfect. When you cut the orientation horizontally, then you may see this section. Each ellipse indicates the area we perceive as the same color. We call this as a color tolerance because we are tolerant of a color difference within each ellipse. At a glance, the ellipses look similar, but in fact, they are getting smaller. They are getting smaller when they reach to the center. This means that we perceive the color difference by the little change when the colors are achromatic, which means grayish. On the other hand, we are less sensitive to the difference of vivid colors. The ellipses are getting bigger when colors are becoming more vivid. Another uniqueness of the CED is that the hue array resembles how the human vision system processes the color signal. The 3 LAB axes of the CED corresponds to the 3 opponent channels. In particular, the red-green axis is right angles to the yellow-blue axis, which is realized here. Red-green axis is right angles to the yellow-blue axis. The A and B coordinates are applied to some other color systems, for example, the NCS. It takes an advantage of following how the human perceives the chromatic characteristics of colors. The NCS will be continued in the next video lecture. Thank you for your attention.